Assalamu alaikum dear students. This is Miss Aisha Anwar from Biotic Academy, and I would like to welcome you all in a new chapter of O levels biology, that is biological molecules. And today we are going to start with the explanation of this important topic. In this topic we are going to study about the components which are basically there in our body, which are making up the cell structure, our bones, our muscles, our mass, our genetic material. These are the chemical elements these are the chemical compounds which contain some certain type of chemical elements which make up these compounds, and these compounds together make up the body of the living organism. We'll be studying about their elements, their monomers, and how they form complex structure, and also along with this topic we'll be discussing about the test which we perform in order to find out the presence of these chemical compounds in the body or in the food component of living organisms. So we'll be starting our topic with the discussion of this macromolecules. Basically we have two types of molecules in the body of living organism: micromolecules and macromolecules. Micromolecules are smaller molecules which are simple, and macromolecules are the larger complex molecules which are complex structures which are making the complex part of our body mass. So first of all we'll be discussing about this component of our diet, that is carbohydrate, and the elements which makes up this carbohydrate. As we know that carbohydrate is the source of energy for living organism. So basically carbohydrate is a macromolecule, a giant molecule, a complex structure which is basically being formed by the combination of these three elements: carbon, hydrogen and oxygen. The monomer or the simplest form of carbohydrate is simple sugar. So we take carbohydrate in our diet in the form of rice, potato, pasta, cereals. These are all the different sources of food which provides us carbohydrate, and after the process of digestion the cell receives simple sugar. That's glucose which is being utilized by the cell to get the energy out of it. Now if we talk about carbohydrate exists in different forms in the body of living organism, and the structure which they form is basically called polysaccharide. Polysaccharide is basically a chain structure of simple sugar. When different or same type of simple sugars join together to form a long chain, that is called polysaccharide, and if it is in a simple unit then it is called as simple sugar. So simple sugar when joined in the form of long chain they form polysaccharide like this. So polysaccharides are like this and simple sugar is like this, simple unit which is basically formed in the body of the living organism after the process of digestion. So polysaccharides, in the form of polysaccharide we have different examples in the body of living organism, like starch, like cellulose, like glycogens. These are all the different examples of polysaccharides. And what are polysaccharides? Polysaccharides are basically the forms of carbohydrate. Carbohydrate may have this monomers or may have this polymers. So monomers example is the simple sugar, and polymers example of polymers form is basically this polysaccharide. And different forms of polymers are basically present in the body of living organism, like this starch or glycogen or cellulose. Now the second comes lipid. Lipid is basically fats and oil which we need in our diet in a small proportion, because they supply a considerable high amount of energy as compared to carbohydrate. You can see that in carbohydrate we have carbon, hydrogen and oxygen, and in lipids we also have carbon, hydrogen and oxygen, but the proportion of these elements is different in lipids as compared to the carbohydrate. And because of this difference in proportion of these elements, it is basically rich in energy. The monomer of lipid is basically fatty acid and glycerol. Together they join together to form the lipid molecules, and the polysaccharide, or you can say the polymer of the lipid or you can say the complex form of lipid is basically this lipids. And the example of lipids is these fats, oil and waxes. These are all containing these monomers, fatty acid and glycerol, with a proportion of carbon, hydrogen and oxygen, and the proportion of energy released by the lipid is quite high as compared to the carbohydrate. Now the third one is protein. Protein also contain carbon, hydrogen, nitrogen, oxygen and sulfur. So you can say there are two new elements here, that is nitrogen and sulfur, which are present in protein. And the monomer or the simplest form of protein, which basically we get after the process of digestion, is amino acid. This is the form of protein that is needed by the cell. So amino acid is basically the simplest type of protein, and when the long chain of amino acid join together, they form polypeptides. So again, we have different types of polypeptides in our body, in the body of living organism. The example is insulin. Insulin is one of the example. There are many other polypeptides in the body of living organisms. The last one is nucleic acid. Nucleic acid are the compounds which basically makes up the genetic material of our body. And nucleic acids also contain carbon, hydrogen, oxygen. Along with it, it also contains phosphorus. Phosphorus is present in the nucleic acid, and the monomer of nucleic acid is nucleotides. These nucleotides again makes a long chain to form the nucleic acid, and there are two forms of nucleic acid in the body of living organism, that is DNA and RNA. These both are nucleic acids. So here I have given you a brief information about these types of macromolecules present in the body of living organism, the elements which makes up these macromolecules, and how the simplest form of these macromolecules converted by the chemical enzymes in the body into the simpler form, that is simple sugar in case of carbohydrate, fatty acid and glycerol in case of lipids, protein in the form of amino acid, simplest form is amino acid, and nucleic acid contain nucleotides. So these are the monomers, and their long chain form the polymers. Here you can see these polymers, and their examples are here. So this is the brief information about these macromolecules of the living organism. Now if I want to find out about the presence of these macromolecules in the body of living organism or in any food sample, so how would I know that these particular macromolecules are present there or not? So for that purpose, we have certain tests which we perform in order to find out the presence of these macromolecules in the food sample or in the fluid of the body. Like for suppose to check the presence of reducing sugar in the body of a living organism, we can perform a test that is called Benedict test. Why it is called Benedict test? Because the reagent used to test for the presence of reducing sugar or the simple sugar, the reagent that is used to perform this test is basically called Benedict. So how we are going to perform this test? We are going to perform this test by taking a 2 cm cube of test solution, means we are going to make a solution of glucose in water, and then we will put this test tube into the beaker, which is basically having a warm water, or we can make a water bath. Before putting it into the water bath, we will add few drops of Benedict solution in the test tube. So the Benedict solution, when it is kept in warm water or in water bath, it will start changing color, and that color change you can see here in different transition of color can be seen, and these color changes are basically there in order to give us an idea about the concentration of reducing sugar present in the food sample. For suppose if the color of the test tube appears blue, that shows that it doesn't contain any glucose in it. So blue color show the absence of glucose into it. Now if it appears green or yellow ppt like this, greenish yellow, then that shows that it contains traces, a very little amount of reducing sugar into it. If it appears orange, that means it contains a moderate amount of glucose into it, and brick red color shows that it contains a large amount of reducing sugar in it. So see, in this way it's very interesting test that where you are going to see different beautiful colors which are basically reflecting the amount of glucose present into a food sample. So this test is called Benedict test, and it's a very important test with reference to the examination, that you will find this test in many questions, and they will keep on asking in different scenarios about this Benedict test. So it's very important with reference to the examination. Now the next test is Biotic test. Biotic test is basically for the check, in order to check about the presence of protein in the food sample. So Biotic test, why it is called Biotic test? Because the reagent used to test for the presence of protein is called Biotic. So in this test, whatever food sample we want to check for protein, for suppose I am taking solution which is containing egg albumin, or you can say egg powder, that is rich in protein. So I will dissolve egg powder into water, and then I will put few drops of Biotic reagent into it. The color will change and it will show purple color. That purple color shows the presence of protein into that solution. That is showing the positive Biotic test. But for suppose if I am taking any food sample which is not having protein into it, and I am adding Biotic reagent over there, the color will appear blue. Why it appear blue? Because the color of the Biotic reagent itself is blue. So it will appear blue color in the test tube, that show that protein is absent in that particular food sample. So in this way we will get to know about the presence of absence of protein in a food sample. So these are very important tests for the examinations, and I hope you are understanding this explanation and enjoying my videos, and you have subscribed my channel. This is Ms. Aisha Anwar from Biotic Academy. Take care.